More genetics, you say? Aha, coming right up. All right, so we're in section 2 of genetics, okay? So section 1 kind of went over the basics of genetics, some of Mendel's findings, and today we're going to jump right into some more lovely, lovely components of genetics. So let's start off with something called codominant alleles. Okay, so as I go through some of these things, I'm going to give you guys examples of what these are, and you're going to notice that some things definitely sound similar, but there's ever so slight differences. But for the most part, you're going to see that there's a lot of similarities in some of these concepts we talk about. So codominant alleles, basically when codominance is present, it's usually in a heterozygote, heterozygote, both alleles are dominant and expressed at the same time. So basically, remember, a heterozygote means that you have different traits. Okay? It's not that you have homo is the same, hetero is different. So heterozygote, if we were talking about brown eyes, a heterozygous brown-eyed person would have the alleles big B and little b. Okay? So heterozygote is different alleles. So in a heterozygote, both alleles are dominant and expressed at the same time. Okay? And what that means is, let me give you an example right here. You have, we're talking about good old cows here, right? So the coat color in cattle, this is an example of that, where you can have homozygous, RR, red coat, but then you can have a homo or heterozygous, these are both dominant, and that gives you a roan coat. This would be the roan color. And then your homozygous, these are both dominant, we would give you your white coat. So I'm going to go back one more time to just read this again so that you guys get that kind of understanding of what you need to do. So codominance, in our heterozygote, both alleles are dominant and expressed at the same time. So typically we don't have something like big R, big W. But in this case, we do, and it changes the coat completely to a roan color. I'm sorry, red coat, and then this is roan. I'm pointing at the wrong things. Hello! So that would be an example of that. And then another example, let me show you this example of these moths. That basically you have kind of this black spotted moth with a white spotted moth, and when you have that codominance, then you end up with kind of a little bit of both. You end up with kind of this gray looking moth still with white spots. So I want you to kind of think that it's a little bit of a kind of smash of both of those, and that's what it gives you. Okay, so codominant. So you have both of those traits that are dominant. All right, let's move on to another thing. Patterns of inheritance. Okay, we're going to talk about some different patterns of inheritance. So here's a couple of them. First, complete dominance, which is something that we've talked about before. In complete dominance, the heterozygous individual, the dominant allele is expressed. The recessive allele is present but unexpressed. So this is something that we kind of mentioned in section 1, where if you have brown eyes, yet your alleles are big B, little b, that little b typically is meaning you have the recessive trait for blue eyes, but it's not showing up because you have what we call complete dominance. So again, in the heterozygous individual, only the dominant allele is expressed, and the recessive allele is present but not expressed. So here's an example, something that we've kind of seen similar examples to in the last section. So you have a tall plant, which is big T, and you cross it with a short plant, little t, all right? And what you get in the offspring, which if you did a Punnett square and you were to cross multiply these two, you'd get all of them big T, little t, which big T is dominant, okay? Complete dominance, so all of the offspring would be tall. Okay, that's showing that that's complete dominance. Now, if you were to cross a heterozygous plant, big T, little t, with another heterozygous, then we got a different story. Okay, so that you'd have to do a Punnett square to see what you get for that. But here's what happens if you cross the big T with the little t. Okay, those little t's may exist, those recessive traits exist, but they're not showing up because you have that complete dominance. Okay. Alright, let's talk about incomplete dominance. So, kind of the opposite. One allele is not completely dominant over the other. Okay? And you have to see, you have to know it's not exactly the opposite, but it's kind of an in-between mix. So So incomplete dominance means that the dominant trait isn't always fully dominant, which is kind of weird. So here's an example that'll kind of bring it together. So the example I have is with hair, curly hair, okay? So curly hair, super, super curly, straight hair. If you have what we call this incomplete dominance, like up there, then basically if you cross these two, you get wavy hair, okay? So basically one allele not completely dominant over the other, so straight hair isn't going to be completely dominant over curly hair, so that this person's going to get straight hair, and vice versa. Curly hair isn't completely dominant over straight hair, so you end up with wavy hair, like I have. Kind of a pain in my rear sometimes, but nonetheless, that would be an example of incomplete dominance, okay? So here's another example, okay? Think about flowers. We call these Japanese 4 o'clock plants, and you'll hear that actually a lot because a lot of genetic experiments were done on these Japanese 4 o'clock plants. You have big R, which produce red flowers. You have big R, big W, which produce pink flowers. And you have big W, which produce white flowers. So a cross between these, and you get kind of a mix, right? Red and white make pink. So here's a picture of it. Let me zoom out just a little bit so you can see it. Okay, so you have your plant here, the red plant, which is big R, and your white plant, which is big W. And you cross them, and again, here's a Punnett square, and you cross multiply, and you get big R, big W for all 4 of the offspring. And big R, big W is kind of a cross between these two, okay? So that's your incomplete dominance, okay? So there's another good example. And my last example, oh, lovely monsters. I had to use monsters. I found this, I thought it was kind of cool. Or aliens, should I say. They look like aliens more than monsters. So you have an alien with 2 horns, okay, little h, and you cross that with an alien with no horns, which is big H, okay? And when you have that, you get big H, and you get 1 horn, kind of a cross between, right? So it makes sense. Okay, so let's move on. So how about incomplete dominance in human traits? So we've talked about kind of some random things. We've talked about flowers. We talked about aliens. But what about humans? Okay, incomplete dominance in human traits. The first example we have is sickle cell. And I'm going to use sickle cell anemia or sickle cell disease for actually quite a few examples because it does pertain to many different types of genetic variations. So when you have a homozygous recessive, that person will have sickle cell disease. Homozygous, meaning 2 of those alleles, okay? But it is a recessive trait, so obviously like blue eyes, you have to have both for it to be the full-blown disease. But the sad thing is, even if you're heterozygous, meaning if you carry 1 of those recessive traits, you have a mild sickle cell trait, okay? So you have some of those things that happen when you have sickle cell anemia. So I want to explain to you a little bit about sickle cell anemia because you're going to hear about this a little bit more as we talk about some other things. But sickle cell anemia is a genetic disease where basically the hemoglobin is abnormal in some of your red blood cells. So here's a normal red blood cell. Looks like kind of a disc, like a donut with not a hole. And red blood cells are very important because they carry around oxygen, right? They carry oxygen to all the different cells in your body. Well, all of a sudden you might have this abnormal hemoglobin in some of your red blood cells, and they turn into this sickle cell shape, okay? And think about it, capillaries are the smallest blood vessels you have in your body. They're only 1 cell thick. They're microscopic. So think about a cell like this trying to get through this teeny little blood vessel. It's going to get caught. It's going to cause clots. It's going to be very painful. I actually had a friend who had sickle cell anemia, the full-blown disease. And it was really sad because anytime she'd have kind of what she would call a bout, where she'd have a lot of these sickle cells kind of form and basically start to clot, she said it was incredibly painful, and she'd be out of school for like 2 to 3 weeks. Now, obviously, there are things that you can do, and I don't know the exact treatments, but you know there is some medication that you can take to try to kind of break up these little clots and so forth. But it's kind of a devastating disease to a lot of people, and we'll talk about kind of who it tends to target and why. Okay, so we'll come back to that in a little bit. The next thing is Tay-Sachs disease, and let me kind of make this just, that's too big. Okay, so Tay-Sachs disease If you're a homozygote, meaning you have two of those same recessive traits, very serious symptoms. Okay, you're unable to metabolize certain lipids due to non-working enzymes, and then lipids accumulate in the brain and cause seizures, loss of mental and motor skills, and ultimately will cause death in a few years. So if you are born with Tay-Sachs, it's a really devastating disease, because especially if you're homozygote. So you're going to have all of these basically fats that develop within the brain and around those neurons, and ultimately you're going to lose all of the ability to do everything, and ultimately your lifespan is a very short lifespan. Now if you're heterozygote, meaning you have one of the recessive traits, you may not have those symptoms of Tay-Sachs, but there will be some detectable biochemical effects. Stuff that you can live with. You'll live a nice, long, healthy life, but there are going to be some effects from that recessive trait, that one allele that you have, okay? So those are two of the things that are basically incomplete dominance within humans, okay? So they still kind of show their colors, but not fully unless you're homozygous, okay? So make sure, if you guys do not understand the terms homozygous and heterozygous, ask me in class and I will definitely go over those again. But homozygous, homo is two of the same. Hetero, different, okay? Remember, we're talking about like the genotype. The genotype would be, say for, again, I always use the easy example of brown eyes. If you have a heterozygous brown-eyed person, that genotype would be big B, little b. If you had a blue-eyed person, you'd have little b, little b. That's homozygous. Heterozygous, big B, little b. Two different alleles, okay? So make sure you guys understand that, because you'll hear that a lot. So now let's talk about multiple alleles, okay? Multiple, and by the way, we will come back to sickle cell, because I've got more stuff to talk to you guys about that. So multiple alleles is having more than two alleles for a trait, because usually we're dealing with, just say, maybe two different colors, like red flowers versus white flowers. But sometimes you have something called multiple alleles. We're dealing with more than two alleles. So in this case, human blood type and sometimes even eye color are good examples of multiple alleles. Because remember, it's not that we're just dealing with brown-eyed and blue-eyed. Look at all these different colors that people have. I would say I'm probably, I don't know where hazel is, maybe this color. So really you have more than just the two brown-eye, blue-eye, and then kind of the mix. You may have, kind of that green allele in there. But a better example to really show you guys is actually blood types, because I can't really show you as well as I can show you the blood typing example, okay? So first, I'm going to show you the three alleles would be A, you guys know blood types, A, B, and O, although there are four blood types, A, B, and O. But the three alleles are A, B, and O, okay? So A and B are codominant, where O is recessive. So meaning, remember, someone can be type AB blood, and because A and B are codominant, they can basically show up together, okay, and be a different blood type even though they have both of those alleles, okay? O is recessive, so the only way that you're going to have type O blood is if you have two of these O recessive alleles, okay? So let's look at this diagram here. Let me make it a little bit smaller so you can see. The ABO blood system, okay? So I want you to know, and I'm not going to go over this in depth because there's a lot to this actual picture here. You have blood types, type A, type B, type AB, and type O. So the alleles, or the genotype that you could have for type A, would be A and A, or A and O. Because O is recessive, it's not showing up. Remember, A is dominant over O. So type B blood, you could have BB and BO. Type AB blood, you can only have AB. And type O, you'd have to have O and O in order to have type O blood. And some of you guys might know this, but type O blood is actually the most common. Okay, I'm type O. AB is the least common. Okay, so again, I'm not going to go into this too much, but the cool thing about the blood types is basically what these surface proteins do. They are telling all the other cells, especially red blood cells, what type they are. So you have these little markers on each of your cells. So if you're type A blood, you're going to have these little A markers. And if you're type B, you're going to have B markers. If you're type AB, you're going to have both of those. Type O has no markers. So in your plasma, you have these antibodies. And this is where the whole transfusion comes in, where You could never give someone with type A blood type B or type AB because there are these B antibodies that will attack those cells. So if you have any B markers on the cells, those B antibodies are going to attack it, clump together, and you die. Okay, so not quite that drastic, but same with if you have type B blood, you're going to have A antibodies, so therefore you cannot get type A blood, and you cannot get type AB blood, because that will clump together and you'll die. But type O blood has no markings, right? So O can give to A, can give to B, can give to A, and can give to AB. We're called the universal donor. We can give blood to anybody, but we can't receive from anybody except for O. So that could be a whole new conversation in itself, and actually we'll talk a little bit more about that next semester. But ultimately know that there are 3 different alleles, okay? We're dealing with A, B, and O when we're talking about our blood typing. Okay, so enough of that. I probably went into that way too much. So here are some of the genotypes and the phenotypes. So you can have AA, which is homozygous dominant. You're going to have type A blood. BB, homozygous dominant again, type B blood. AO, heterozygous, type A. BO, heterozygous, type B. AB, which is codominant, so that's going to give you type AB blood. And then OO, homozygous recessive, gives you type O. Just another way to look at it, okay? So there's multiple alleles for you. All right, I think we're moving on, getting close to the end here. Epistasis, something kind of interesting. Epistasis is where one gene affects another gene, okay? Epistasis, I had to make that a little bit bigger in case you couldn't see it. Okay, one gene affects another gene when one gene blocks or masks the expression of another gene, okay? So it's really interesting that, take for instance, one of my big examples is albinism, which I'll get into in just a second. But here is showing some dogs, okay, and I'll explain this term again, but let me show you these pictures. So a dog with alleles B and E is black. Interesting. A dog with alleles little b, and E is brown. And a dog with little e is yellow, regardless if it's big B, little b. So the yellow lab is showing that basically this little e is basically masking any of the other traits that that dog has. So it's going to be yellow no matter what. So maybe it has a big B, maybe it has a little b. So it has some of these traits that make these labs black and brown, but because it's little e, it masks everything. So my favorite example, let me go back to it, is albinism. Okay, if you guys have ever met an albino, I actually went to school with a girl who was albino, and what that is basically they have no skin pigment whatsoever. That means they have no color in their eye. In the iris, there's no pigment there either. That's why they have red eyes. And the reason why they're red is because that's where you can see actually the blood vessels. Not that you can see the little blood vessels, but it's red because of the blood vessels within your eye. So if you didn't have any pigment, you would have red eyes no matter what. So these people, good pictures, rat and 2 girls that are albinos, they may have traits for other things. They may have traits for freckles, like I have freckles everywhere. But because they have this albino trait, it masks everything else. They may actually have the trait to be super tan, but because they have this one trait that says, I'm going to mask everything else, you're an albino. Okay, so that's what epistasis is, okay? So one trait will mask everything else. Regardless of whatever else you have, that one trait is going to basically overpower everything else, okay? That's epistasis. Now the last thing I want to talk about, how can the environment affect gene expression? So it's kind of weird, but actually I think that's going to come into play even more these days because our environment is changing so much with so many chemicals and so much junk that our bodies are being susceptible to, or our bodies are susceptible to, that I think it's going to affect even more the gene expression. And you know, that could be a topic of conversation down the road. I'm sure that people have attributed the higher levels of autism in children to some of the environmental effects, but nonetheless, and I like this little puzzle piece down here, okay, because this puzzle piece is, let me get it a little bit bigger here. So you have a piece of puzzle with kind of this environment and then the genes, and I think this is actually a really good kind of image to represent what we're going to talk about. So moving on, one gene, many effects. Let's talk about sickle cell anemia again, because this is a great example. So sickle cell anemia, I told you, is where you have this genetic defect where some of your red blood cells turn into this sickle shape. I can't talk. I always make these at night, and then I'm like, blah, 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 blah. So these sickle cell cells, they basically do not go through blood vessels very well, okay? So the interesting thing behind sickle cell anemia, and this is just kind of a little table showing you that someone with homozygous recessive individual will have abnormal hemoglobin, sickling of red blood cells, and it can go to all these different things, and this is just showing you some of the different effects of sickle cell anemia. But here's the interesting thing. Most of sickle cell anemia occurs in the African American population. And the reason is because that population, when you guys, if you've ever known anybody that's gone to Africa, you know that they have to get malaria shots, and they have to kind of prepare their bodies for malaria, because malaria is it's just a predominant disease over there because the mosquitoes are carrying it. So it's interesting that because of that environmental effect, that a lot of African Americans have developed this genetic disease, but because of that disease and because of their abnormal hemoglobin, they are immune to malaria. So it's kind of a negative and a positive. You know, you have this disease that can give you a lot of these side effects: skull deformation, weakness and fatigue, impaired mental function, poor physical development, pneumonia, rheumatism, paralysis, and such. You know, not everybody has all of these, but it can get to that point depending on how serious it is and if you're not kind of taking care of your body and so forth. But it does protect these people from malaria. So there's kind of that genetic environmental component, kind of how your body can kind of somewhat adapt, but kind of adapt in a weird way, in kind of a negative way, genetic defect, okay? Interesting though. So that's a little bit more behind sickle cell anemia. Now, the last thing I really want to get into is genetic disorders, and we can talk endlessly about this. There are so many genetic disorders out there, and I really ask you guys that take an interest in that. There are people that have some severe disorders because of some minor things in their genes. So I'm going to show you this video. We're not going to watch the whole thing. It's pretty long, but I want you guys to see just how many genetic disorders are out there and how devastating some of them can be with just these small minor changes within their gene code. Okay, so watch this and just kind of take this to heart and be thankful and appreciative that you most likely are not suffering from some of these things that these people have to go through. Okay.
Somewhere on chromosome 7, there is a gene that has 6,129 active letters in it. This gene strings together 1,480 amino acids into a protein whose function is to allow salt to penetrate various membranes in the body. Little Susan has a tiny error in that gene in her 7th chromosome. 3 letters are missing, and that causes the protein that Susan's genes manufacture to be ever so slightly misshapen. And although the difference in the shape is tiny, the consequences are huge. This particular protein does its work in the linings of the lungs. By inserting itself into the plasma membrane, the protein makes a passageway for the salts to enter and leave the cells. But if the protein is not exactly the right shape, then it cannot transport the plasma membrane and therefore cannot do its job. Then salt gets trapped inside the cell, and that causes the cell surface to become sticky and covered with thick mucus. This results in wheezing, breathlessness, and the persistent cough to expel troublesome mucus. This is cystic fibrosis, and it is the result of just 3 missing letters on chromosome 7. Perhaps even more tragic is Tay-Sachs disease. Tay-Sachs begins at one infinitesimal spot on the DNA ladder when just 1 letter goes wrong. This mistake comes down to just 4 atoms. That's it. But the error in that gene creates a problem in this protein, which is supposed to dissolve fatty materials in the brain. But now the protein doesn't work. So fat builds up, swells the brain, and eventually strangles and crushes the critical brain cells. Infants with Tay-Sachs disease appear to develop normally for the first few months of life. Then, as nerve cells become distended with fatty materials, a relentless deterioration of mental and physical abilities occur. The child becomes blind, deaf, and unable to swallow. Muscles begin to atrophy and paralysis sets in. Hard to imagine that just 4 wrong atoms change just 1 letter out of 3 billion. And this is the result. When genetic diseases run through families, then we know the mutations in the genes that cause these diseases are being inherited. Here is an example. In a normal gene, this is the sequence, ending G, T, A, G, C, A, G, T. But sometimes the gene is missing 2 of the letters. This new configuration is a mutation that can often lead to breast cancer, and it is passed from generation to generation, causing the potential for disease to be passed down as well. The U.S. government maintains a website with much of the current information about genes, chromosomes, and proteins. In the section labeled Chromosome Viewer, you can select a chromosome and see how many base pairs or letters are on the chromosome. When you get to the web page for each particular chromosome, you can see a list of traits and disorders associated with genes on that chromosome. Let's enlarge chromosome 7 that we talked about earlier. As you can see, the gene that causes cystic fibrosis is here. On other pages, you can see genetic disease profiles. Here is the cystic fibrosis profile. You can even examine specifics about some genes. Again, this page details genes associated with cystic fibrosis. How can we fix genes that are broken? Gene therapy is a new and promising technology. Consider a disease like SCID, which is an autoimmune disease.
Okay, I'm going to stop us there in that video because we're going to get into gene therapy in just a little bit. So, the last thing I want to talk to you guys about is sex-linked traits. By the way, there were some things mentioned in there about base pairing. We'll come to that. So if you guys are like, A, T, G, what is that? Just know that we are getting to that, probably the next section, I believe. So hang tight. You'll understand that in just a moment. So sex-linked traits, there are some traits that basically are found on the sex chromosomes, okay? Sex-linked traits are determined by genes found only on the sex chromosomes, which is the X chromosome, okay? So remember, males have XY chromosomes, females have XX. So if there's a sex-linked trait found on the X chromosome, males only have one, so it's going to show up regardless. Where say if females were carrying that sex-linked trait, well, there's another X to kind of help it along and mask that, okay? So basically, anytime you have a sex-linked genetic disorder, it's going to be with males. And the best example is color blindness, okay? Color blindness happens to be a sex-linked trait that's found on the X chromosome. And therefore, in the females, it does not show up because we have 2 of those, but males only have 1 of those. So therefore, males typically tend to be more colorblind. Now, I'm not going to say females are not, because if females have basically the sex-linked trait on both of the X chromosomes, they will be colorblind as well. But no matter what, if it's found on the X chromosome here, a male will be colorblind. So here's just kind of one example of a colorblindness test. This is red-green colorblindness. So basically, hopefully you can read all of these numbers in here, and actually I believe that's a letter, yes. And but people that have red-green red-green colorblindness would not be able to see any numbers. They'd just see all these different squares. Here's some other tests that they might use, or here, I shouldn't say, there was another test that I'm going to show you down below in just a second, but here would be a normal view of, say, this garden, and then somebody with red-green colorblindness would probably see this. Either they do not see those colors, or those colors end up being kind of a different, maybe a different shade of a different color, okay? So sometimes those red and green are completely absent, and sometimes they're like a different shade of yellow or a different shade of something else that they can't see. And the funny thing is my little nephew, I found out, is colorblind, and he's only 5, but they learn to know what is green. They know that they've been taught that grass is green. So they can look at it and they can start to kind of rewire their mind to say, okay, so whatever I'm seeing right there, that's the color green. So they kind of rework that in their minds and say, okay, well, I know if I see that shade of whatever it is, that's green. Okay, so it's kind of interesting. And then one less test that you would see is if you didn't see that S in there, you might want to get checked out. But it's not a big deal. It's not like people that are colorblind can't, function in the real world. So just another example of a sex-linked genetic disorder. Okay, so there we have it for sex-linked traits. And we bring us out to lovely genetics. So we are wrapping up section 2, and then section 3 we'll get into more heavily talking about DNA. So be prepared. But until then, enjoy whatever you are doing at this very moment, and then get to work. Okay, bye-bye.